Exam 3, week from today, right? That'll be fun. Something to look forward to. Should be a practice exam on today's or with today's folder, I guess. That should be open. And so review session Sunday night again. I guess we'll do that same as, if everyone's okay with that. Yeah, I'll have a link on D2L posted by the weekend. Sunday, 7 o'clock. Yeah, hope everybody can make it. If you can, email questions and I can answer them on there. Yeah. Right, so Thursday's lecture is going to be a bit, I was going to do some on it's an e-cigs follow-up, I guess, so I was going to do some on that, and then the second half more review session. So going over the modules for this last section with, I guess, highlighting the most important parts from each drug recovery. So yeah, and then from, I'll, I guess, say on Thursday what portions from the first part of the lecture will be important for the exam. Okay, questions or comments, concerns on anything prior to diving in? Okay, so today is the one day for hallucinogens, which, well, I guess, yeah, all of the drugs we could spend an entire semester on, but this will be a very quick oversimplification for this field of drugs. The two classes of hallucinogens that we'll carry are the psychedelic agents and the dissociative agents. Psychedelic agents are the ones that, I guess, most anecdotally known as hallucinogens. So these are the ones that enhance or amplify thought processes. These are the ones that give rise to extreme levels of euphoria. LSD, also known as acid, mescaline, psilocybin, MDMA, we've talked about a bit, but definitely falls into this group. And then the dissociative agents that aren't talked about that much definitely have a very big part in the history of hallucinogens, but these are the ones that people don't want to talk just due to the fact of what they do with disconnecting thoughts, emotions. So essentially they sever the experience of reality that a person has from, their sensory perception of the world is disconnected from how they see themselves in the world. Essentially living in what feels like a dream and causing a lot of problems because the people actually begin to feel like there is no reality that they're living in, and then they do some really drastic things. But those are the two big different classes. The psychedelics are the fun one, the dissociative are the scary ones. They do have clinical applications, some of them, but we're not talking about that aspect of them in this course. So diving right into the psychedelics, the euphoric hallucinogens shown here, these are agonists at serotonin receptors. We'll talk about the specific ones in a minute. So these primarily modulate serotonergic tone. As you can see, they are similar to a molecule of serotonin. Here, LSD-25, mescaline, psilocybin, and then MDMA all have potency at serotonin receptors just due to the fact that they overlap with the molecule of serotonin that all comes from tryptophan, right? So some of the sources of these, the ergotamine is a natural plant-derived compound that is an analog of lysergic acid. And interestingly enough, people know this or not, Salem witch trials took place a couple hundred years ago, Salem, Massachusetts, and apparently archaeologists or anthropologists that have dug or excavated some of the sites that were part of this town in Salem found that their wheat stores had actually been tainted by these ergot fungus that produce ergotamine. So this is a pretty potent hallucinogen. And the thought is that with this contamination, what they thought may have been happening is that the wheat sources in this town were tainted by these ergot fungi that started to grow on the wheat, and then the people would ingest these and start tripping out and accusing everybody of being a witch, and then panic happened. And whether or not that's true or not is yet to be seen, but that's one of the explanations. Sources for mescaline come from peyote cactus, usually the buds of the flowers from the cactus, and then psilocybin grows. And these multiple different types of mushrooms. So these are the fun ones. When it comes to hallucinogens, the DASC scale is used for evaluating the symptoms that you experience. So dimensional altered states of consciousness, or DASC rating 5 because it fits into the DSM-5 framework. But the different types of parameters that are evaluated are broken into perceptual alterations, anxious ego, oceanic boundlessness. So the way that the scale works is the people, when undergoing some hallucinogen, they will rate from a scale on 0 to 70 the intensity of each of these different types of hallucinogens, imagery that is happening, a blissful state, experience of unity with people, or in the anxious ego category, disembodiment, anxiety, impaired control and cognition. So any of these different parameters a person is asked to evaluate on a scale of 0 to 70, where they are, and you can see the different types of hallucinogens have different patterns. So on the left here, we're looking at 3 different doses of psilocybin. So these primarily work in the perceptual alteration and then somewhat in the oceanic boundlessness. So here's our euphoria here, but then here's our hallucinations over here. So as you can see, with increasing doses of psilocybin, you have increasing perception in these different parameters. Looking at the right side, ketamine is a dissociative agent. So the more on the anxious ego, definitely the disembodiment, impaired control and cognition are heavier here. Surprised to see the experience of unity, but ketamine isn't as bad as some of the other dissociatives. So anyways, but the bottom line is that's how these are evaluated in more of a clinical setting. And as you can see here, this is one of those depending on where you read type of thing. As you see, the different categories are changed here, but some of like the disembodiment is now in this group rather than with that altered ego group from the previous slide. But bottom line is the DASC rating scale is used for evaluating the effect of hallucinogens and all of these different types of parameters. As you can see in this example, they're evaluating actual electrical stimulation in parts of the brain, and then different types of LSD, MDMA, and psilocybin, so different hallucinogens there, and you can see where they rate on this scale. Okay, so LSD itself has a very interesting history. Albert Hofmann is the chemist who is given credit for synthesizing this agent. He was studying, as it shows here, the ergot alkaloids to try and see if they could come up with a compound that would help treating psychiatric disorders as well as drug addiction. So many of these drugs, whether they're hallucinogens or stimulants or anything, are developed as a means of treating addiction to other drugs. But they say he accidentally ingested a small quantity. The stories that I heard actually is that he brushed his hands, medicinal chemists and pharmacologists and people that used to, were well, in the earlier days of these fields, when they would actually synthesize a compound, things were much different than today, right? Nowadays you have very strict regulations of how you're going to test things, especially when you go from biochemical style tests into actual animals. You have to get investigational review board permission to do any work with animals, especially if you're working with primates of any level. But back in the day, as people say, when people used to, at Albert Hoffman's time, 50s and prior, when people would synthesize a compound, I mean, you would honestly just have your isolated compound on your desk and people would just take it. Is how people would test it. And if your heart started racing in that, you know, you would have some kind of adrenergic toggling. If your blood pressure was depressed, you know, it's some kind of muscarinic toggling. So that used to be the way it was done. I don't know if that's what exactly Hoffman did. The story that I heard is that he had tried to move the sample aside as he was writing in his notebook. It brushed on his hand, and LSD can be absorbed through the skin, and he started on an amazing trip, right? He had this incredible experience, went home, told his wife about it. So here it says as little as 20 micrograms can produce an experience that lasts more than 12 hours. That's pretty rare. Standard dose for LSD is 150 micrograms. Usually about 6 hours, they say, and I just wanted to include that because the story then goes that Hoffman went home, told his wife he had the best day at work ever, he had the most amazing experience ever, he found this compound, blah, blah, blah. Went back to work the next day and ingested 4 grams of LSD. So 150 micrograms is a standard dose, 4 grams would be 4 million micrograms. And that's way saturating now. But the interesting thing with that, and that's, yeah, we love to tell that story because it's ridiculous amount he would take. But the thing with LSD, it's not a terribly toxic compound. And once you saturate your brain and receptors in that, you just pee the rest out. So it wasn't like he's under any, you know, we didn't lose him for 60 years in some corner of the world because he had lost his mind. It's just kind of an odd story. But he did actually ingest 4 grams of LSD, and that's amazing in and of itself, but whatever. Back to the story. So there was a lot of interest in these through the 50s and 60s. Lots of papers punished. However, when there was a massive recharacterization, reclassification of a lot of drugs, hallucinogens went on to Schedule 1, and then all of that research really just kind of crashed, and we lost a lot of it because people just threw it out, wanting to not acknowledge the state of, or what these drugs can potentially do. So anyways, that's an interesting anecdote about LSD. Here is a picture, I guess, clades of all different GPCRs in our body. Everything that has an orange or a green are the receptors that LSD hits, primarily within serotonin. So yeah, all biogenic GPCRs here, biogenic amine GPCRs. Knowing all these isn't that important. So here's our glutamates, rhodopsin, that's how our eyes see here, olfactory, how we're gonna smell. It doesn't matter. Point is, these are all the ones that are hit by LSD, primarily in the serotonin world, but that's not important. What's really important are just these 3 here. Please know 5-HT1A and 2A and 5-HT2C. Just those 3 are the primary ones you need to know for this class in terms of where hallucinogens will target, okay? Been over this before. Serotonin comes from tryptophan. What will be important too for these 5-HT1A receptor is coupled to G-alpha I, and that causes the decrease in cyclic AMP that we've seen before. Here's a one odd one we haven't talked much about. G-alpha Q is for the 5-HT2 isoforms A and C. So Q is similar to S in that it's going to increase PKC activity, usually through activation of adenylate cyclase that can increase cyclic AMP levels, turning on PKC. And then the beta gamma again can activate calcium channels, inducible calcium channels. So remember that for 2A and C, it's coupled to GQ, 1A is coupled to GI. Where they're at in the brain is also important. So the 5-HT2A are primarily, well, both of the 2A and C are going to be PFC and thalamus, right? So thalamus and PFC, C for both of those, with the 5-HT1A primarily being located in the locus coeruleus, okay? So the RAFE neurons will project out into the cortex and the thalamus. Those are primarily serotonin. Locus coeruleus, as we've covered before, are norepinephrine, projecting into very similar areas, and they're involved in different aspects. The serotonergic neurons are involved in the sensory information, and then locus coeruleus will be responsible for attention. These are our novelty detector style neurons. Okay. So here's normal functioning of serotonin. I believe you've covered this before. Presynaptic nerve terminal, postsynaptic nerve terminal. Serotonin, 5-HT, gets synthesized from tryptophan, stuffed into synaptic vesicle, released out into the synapse, can bind to receptor, 2A, 2C here, both GQ coupled, increasing PKC activity and intracellular calcium, okay? Normal cycling, right, and then the serotonin will be brought up by the transporter SERT, and then it can be broken down by MAO or repackaged by VMAT. With LSD on board, you actually have LSD can just bind to the receptors, working as an agonist at these receptors. And then a bit more about how this would work is shown here. Since this would be thalamus, right? You have LSD working on 5-HT2A here. So this would be here. That's going to be releasing glutamate in the cortex for our output there, all right? A bit more specifically, since so much of what we discuss here is activation of the reward pathway through the drugs we're talking about. Here we're really talking about increase of serotonergic tone in specific areas of the PFC. More specifically, cortical pyramidal neurons are what house the serotonin receptors that allow for this euphoric experience when you have LSD, or in this instance, psilocybin bound, okay? And one of the ideas that is gaining traction now, it's not just the ability for these agents to cause increased excitability in these neurons, it's also a potential for developing or promoting the plasticity that we're really learning about here. So these neurons can actually start to continue to diversify, if you will, or continue to grow with this type of stimulation, all right? So the figure here is just showing what we've talked about before with binding of an agonist to a receptor and then turning on different activities in the side. What I'm really focusing on here is the ability to, of these hallucinogenic psychedelic agents, to turn on serotonin receptors and then have an effect on the actual maturation of the neurons in these areas, not just the development of the mental and emotional states that these drugs cause, okay? So they're remodeling, not just causing some short-term sort of euphoria or experience, okay? Tolerance can develop here, should not be very surprising as this happens with repeated administration of most of the drugs we talked about. So as we're looking at here, response to LSD, if you're pre-treating with either saline or LSD, so pre-treatment is in white here, post-treatment is in gray here. So if we pre-treat an individual with saline and then we give them LSD, they have a full experience on the LSD, okay? But if we pre-treat with LSD and then afterwards treat with LSD again, we have a much more decreased experience than initial due to the development of tolerance. And this can actually be shown on the molecular level, or actually binding in these areas while using an iodinated LSD analog, well, it's not analog, but it's a radio-labeled LSD molecule, and finding where it binds to serotonin 2A receptors. Looking at all these different areas of the brain, you can see chronic pre-treatment with vehicle, which would just be saline, you have a certain amount of binding. Chronic pre-treatment with LSD, you can see all of the different areas of the brain have a drop in the amount of iodinated LSD binding, meaning that you have less binding of this type of LSD at these areas due to the downregulation of these receptors in this area with pre-treatment of LSD, okay? So tolerance here happens. It also happens as a actual physical explanation that you have downregulation of the receptors, so you have less binding of LSD after pre-treatment with LSD. One very interesting thing about hallucinogens is how they don't just change the serotonergic tone in brains, they can actually alter how each individual neuron fires in the locus coeruleus, right? So these are our norepinephrine neurons. However, when treating with LSD, the normal cycle of firing of a neuron would look like this. You know, it doesn't show our full action potential here, but each of these would be an action potential. And as you see, you have the increase, rising, falling, recovery is all in each one of these with a certain washout in between, right? A certain recovery phase. When exposed to hallucinogens, they have clustered firing, it's referred to as. So you'll see instead of these individual action potentials, you'll have a cluster of action potentials in rapid firing, then a longer washout. Cluster of action potentials, washout, or recovery, excuse me. Cluster, recovery, cluster, recovery. So it changes the actual firing pattern in these neurons too. Kind of a black box in this field, but they've seen this pattern of behavior. Okay, so to re-emphasize that in this instance, it's not so much about targeting the reward pathway. We have similar overlap in where we're working in the brain, but because we're really working through a serotonergic tone and not a dopaminergic tone, this isn't really about the standard reward pathway that we've been covering. This is about a different area of the brain. Elevated serotonergic levels can give rise to euphoria, but there's a distinction in between what we've been discussing so far and hallucinogens, okay? So, showing similar to what we've looked at before, here's our VTA. The nucleus accumbens would be somewhere along here, because then here's our dopamine inputs into our prefrontal cortex, glutamatergic neuron coming from the PFC back to this dopamine neuron in the VTA. But our locus coeruleus here, so here's our norepinephrine inputs, and then R for our RAFE nuclei, and here's our serotonin inputs. Here's our pyramidal neurons here in the PFC. So all of our sensory input will be filtered through thalamus, right? And then several different areas prior to sending input to the PFC for some kind of response, right? But what we're talking about specifically is this area here, serotonin input into these pyramidal neurons from RAFE nuclei, okay? One odd aspect of hallucinogens that's specific to this drug is the experience of hallucinogen persistent perception disorder, HPPD, or flashbacks. This is a re-experiencing of some feeling you had while on a hallucinogen prior to this. And it's a very confounding thing because there are people who go through these and there are people who don't go through these. And they don't exactly know why these happen or what the actual molecular basis is of them. However, some of these flashbacks can be so strong that people will avoid wanting to socialize with people with fear that one may happen while you're in public. So very difficult to assess these sorts of things just due to the fact that there's no, we don't understand why some people have them, why other people don't, and then the level of intensity that people go through with these. So they have a really hard time studying them. Potential mechanism explanation for this is a downregulation of the 2A serotonin receptors. But yeah, not a lot of proof behind that. And then one of the biggest things that research community seems to acknowledge, fortunately, but uses that as a confounding factor to this whole thing of why it happens to some and not to others. Looking at a study of members of Navajo community have shown that about 500 subjects that had peyote on more than 100 occasions reported 0 incidents of this HPPD experience. So bottom line is they have had, or there's enough of these experiences recorded from people who have used hallucinogens to know that they exist, but being able to track down the molecular details behind why they happen and what is going on when they do has been elusive to this time. So it's not all fun and games with these. There's a number of different effects that these drugs cause that can be very unpleasant to some. So increasing cardiac output and then other, I guess, nervous attributes, if you will, are one aspect of hallucinogens that really show, or how do I say this? Should determine that certain people should avoid these types of drugs. One of the things that LSD and other hallucinogens has been known to do is triggering psychosis. People who have certain nervous disorders have been shown to have episodes triggered when taking hallucinogens. And it causes just a catastrophic meltdown in the individuals, where they're supposed to be enjoying a euphoric experience, turns into a very dysphoric experience and causes hospitalization in a lot of people. So toxicity in LSD is rarely a physical, overdose, if you will, or something that someone needs to be revived. It's usually a mental or emotional overdose, again, if you will, where people are sent into a state of psychosis that they have a hard time recovering from even after the effects of the drug have gone away. So we covered this before, but I just wanted to bring it back up because MDMA can fit under the, it fits with the stimulants we talked about, but it also fits with the hallucinogens. But just remember the unique aspects of it. It can be brought up into the presynaptic nerve terminal by the serotonin transporter. Big difference with this is that it causes a reversal in the direction of the transporter after it is inside that neuron, causing efflux of serotonin transmitter out into the synapse. So the presynaptic nerve terminal just starts dumping serotonin out into the synapse. Yeah. Okay. One very current, very interesting, but frustrating aspect of MDMA use is its potential use to relieve PTSD symptoms or post-traumatic stress disorder symptoms. The study on the left shows a evaluation from the CAP score, so clinician administered PTSD score, CAPS 5 usually, so also falls within the DSM-5 parameters. But this scale essentially shows that the PTSD score of individuals will drop once on MDMA compared to just a saline placebo. A study just done back in 2021 shows the exact same thing, but this time using 3 different evaluations of PTSD or depression. So here, blue lines are the control, red lines are MDMA treated, and in all 3 instances we had a decrease in the scale or reporting, meaning that the PTSD symptoms improved on all of these people, or in each of these instances. The way this study was set up, you can read the individual details here, but this is a really interesting study because so they have the standard recruitment or enrollment where they're gathering people and then they actually enroll everybody in. Here they had a very unique taper off session. So only people that were able to be to taper off of current medication they were on were eligible for this study. And they had a fairly elaborate tapering off just to make sure they didn't have confounding parameters in this study. So then they would prepare people for this study. They had individual risk assessment at 4 different time points just to make sure everybody was doing okay. But then they would, at 3 different areas, they would dose them with MDMA. And then they had sessions where they would meet together and then go over questionnaires and see how the people were doing. And the results here, this is just a different plotting from the results shown from the prior slide. But here, placebo is on the left and MDMA treated is on the right. And what you can see, 3 different sessions here. The gray and white bars are withdrawal or non-responders, so these are bad results or people that stayed within the PTSD, parameters. Bluish and then reddish is what we would want. So I don't know why I cut it off here. The darkest of the colors is total, relapse. Cured from the PSD symptoms, I guess you could say. And then we have loss of diagnosis and then responders. So the darkening in color shows better prognosis is the bottom line here. And the best is after 3 sessions, you can see roughly 30% of these people are essentially cured from PTSD or remission. That's the word I'm trying to think of. In remission technically. So, but compared to maybe 5% with placebo, this is an excellent result. So and then roughly 80% of the participants versus about 50, but still a good gain in this instance. So, a lot of contention in the field. There's a lot of people that believe this is a great tool for treating this, and then there's a lot of hesitancy to use it, specifically because earlier studies have shown toxicity when it comes to using MDMA. I'd have referenced this a bit when we went over that section before, but hippocampal volume has been shown to drop in chronic ecstasy use. So we're plotting hippocampal volume as a function of treatment, either control or ecstasy, and you can see a huge difference in the volume here. Also, when monitoring the level of serotonin receptor with chronic MDMA treatment, we see a large drop. So this is evaluating the amount of this protein in rats. So these would be postnatal day 37, postnatal day 58. These would be adolescent rats. These would be young adult rats at day 58. And what you can see is that in 5-HT2A, protein levels drop with chronic MDMA treatment in the adolescents versus young adults. The 2C protein level drops. Not so much in the other levels, but the idea is chronic MDMA use can drop levels of serotonin receptor as well as ablate certain amounts of hippocampal volume. One caveat with this is that these studies, this was done prior to really understanding rearrangement and plasticity in brain regions. So as you can see, there's still a large area of hippocampal volume that is still overlapping with the controls, right? The concern is down here with the loss of volume, but the idea or a reinterpretation of this data suggests that these brains may be undergoing plasticity and rearrangement and may not necessarily just be a pathologic loss of actual volume in these treated animals. Okay, DMT, the last hallucinogen we'll talk about. Dimethyltryptamine, Just remember, so tryptophan is converted into serotonin. In the pineal gland of the brain, it's actually made into DMT as well as melatonin. DMT is kind of interesting. It is a very short-acting but much more intense hallucinogen that supposedly has a religious or spiritual feeling to the euphoria itself. And how this works is thought to be not only through serotonin, so here's our 2A serotonin receptor, but other serotonin receptors as well as other receptors in the brain as well. So it's thought to hit more than the standard serotonin receptors. Okay, on to the other side of things. Dissociative agents, again, are the ones that sever the connection with reality and cause a misunderstanding of what is real and what is imaginary or fake. Most popular agent in this space is definitely phencyclidine or PCP. Ketamine is also a dissociative agent, but this has clinical use as a primary anesthetic or induction anesthetic, so it could put people to sleep. And then one of the most common and over-the-counter available, dextromethorphan. This is kind of a morphine analog, but it does have some overlap with glutamate receptor we'll talk about in just a second, which is how these work as antagonists at glutamate receptors. So here's our glutamate receptor. This is also an ion channel, right? So it allows for efflux potassium and influx of calcium and sodium into the cell. PCP can actually bind into this receptor and block it, okay? So it's an antagonist at the glutamate receptor, and that's how they work. So they're also going to tamp down the activity in the brain, but to a much more drastic level than just the standard anesthetics, and then with the overlap with the hallucinations that happen, causes all kinds of interesting things. So PCP specifically will mimic the symptoms of schizophrenia, as says here. So it causes cognitive deficits that occur in schizophrenics, decline in memory, attention, etc. And there are specific drugs, haloperidol, chlorpromazine, that are used to treat specific symptoms. So it doesn't treat the disease itself, but can treat the symptoms. So positive symptoms can be treated with those drugs, but not the negative symptoms or the cognitive deficits. And those look like this. So the positive symptoms of schizophrenia are the hallucinations and delusions, which is really, want to know why they consider those positive symptoms versus negative. The negative symptoms would be anhedonia, so you don't, and apathy, you don't care about anything, you don't enjoy doing things. Avolition, no motivation to do anything. Blunted affect, when you don't smile at all, you walk around grumpy Gus all day. Versus the cognitive symptoms, the memory loss, inability to process social cues or stay focused on anything. So, the drugs used to treat schizophrenia can treat these afflictions, but not these so much. So chronic low-dose treatment of rats produces conditions that mimic schizophrenia, and then treatments using the neuroleptic drugs, the haloperidol, can restore some of these or treat the positive effects again, but not the other effects, and specifically within the temporal and thalamus lobes, but not PFC. So anyways, that's it for hallucinogens. Yeah, let me know if you have questions or concerns.